2020, Day 3, myself, Alice, Conference Executive of Cell Science Conference. Thanks for connecting Day 3. And I would like to introduce Nadia A. Mohamed from National Research Center, Egypt. And our title will be Biocomplicable Amifostine nanoemulsion via expression of nephrin in nephrotoxic experimental rat. Thank you.
Assalamu alaikum. Good morning, everyone.
Good morning.
Good morning, everyone.
Good morning. Good morning. Thank you. Start now or my PowerPoint? Start now or what?
Yeah, we can start now, Dr. Nadia.
I couldn't see my presentation.
Do you want us to share the screen of your PPT?
Yeah.
Do you want us to share the PPT?
I wanna see my presentation.
Okay,
Could I see?
Okay.
Hello? Hello?
Is it fine, Dr. Nadia?
Okay, it's okay.
Thank you.
Bismillahirrahmanirrahim. Good morning, everyone.
Good morning.
Next slide, please. Next slide. Therapeutic is biocompatible amifostine nanoemulsion by expression of nephrin in nephrotoxic experimental tract. Next. As we know, physicians prescribe drugs to prevent or treat disease, but most of drugs are toxic for many reasons. Maybe for genetic predisposition, non-selective action, inappropriate use or administration of this drug. Pharmaceutical and biotechnology companies spend years and millions of dollars to understand safety and inherent toxicity of these drugs. As we know, all the drugs have adverse effects at normal doses. Next. Drug and its metabolites interact with receptors in the body to mediate adverse effects. These metabolites could be detoxified or excreted from the body, but sometimes interact with macromolecules like DNA or some antioxidants such as glutathione or cellular or plasma protein. That lead to the formation of unrepaired or misrepaired DNA, which lead to mutagenic and maybe lead to cancer effect. The impairment of oxidative stress can lead to inflammation and cell death. Next. Kidney is the main target for drug-induced toxicity because proximal tubule cells, PTC, is affected due to their role in glomerular filtrate concentration and drug transport. Many drugs in the market, like cancer drug, antibiotic, immunosuppression, are nephrotoxic and angiopathy. Drug-induced nephrotoxicity can lead to acute kidney injury or chronic kidney disease, which make patient in the major problem for clinician. So the development for less nephrotoxic drug is a challenge due to the fact that prediction of nephrotoxicity during the drug development remains difficult. Next. Proteinuria is a marker for earnest renal disorders and considered risk factor for progression of renal disease. Dysfunction of glomerular filtrate lead to leakage of plasma protein and diffuse of podocyte food process, which detected in nephropathy. Proteinuria or heavy proteinuria could cause nephrotic syndrome. Experimental model, animal model of glomerular show correlation or high correlation between nephrin expression and proteinuria. Nephrin expression is studied in certain acquired kidney disease. Next, please. Next. Uh, Therefore, there are great need to present from new treatment to ameliorate a toxic effect of the drugs, which the patient is forced to take them for a long period or permanently. So this study is elucidated to found a new way to get the treatment without falling in new drug toxicity. Next. Make a nephrotoxic model, we use cisplatin. It is a platinum-based drug which is used as anti-neoplastic agent for the treatment of cancer. But despite of clinical usefulness, cisplatin have a several toxic side effects like nephrotoxicity, hepatotoxicity, and cardiotoxicity. The excessive stress through generation of the reactive oxidation decreases and oxidation defense system are the major alternation in the cisplatin toxicity. Next. Mechanism of cisplatin as the anti-cancer through formation of reactive complexes with DNA lead to the denaturation of DNA and cell cycle arrest. Mechanism of cisplatin nephrotoxicity is different than activity as anti-cancer. The formation of reactive oxygen species and renal tissue damage is the result for this cisplatin nephrotoxicity. The result lead to acute renal failure by induction of oxidative stress, inflammatory cytokine, tubular interstitial inflammation, as well as apoptosis or necrosis of the renal tubule cells. Next, please. Amifostine. Amifostine is an organic thiophosphate antioxidant, which is used as protector for normal tissue in patients with cancer from chemotherapeutic or radiotherapy drugs. This amifostine become active form by hydrolysis by alkaline phosphatase to prevent DNA damage. But active metabolites have limited stability in vivo because it is rapid formation of disulfide from free sulfhydrides, as also it should be used in high dose to realize a protective effect. This high dose lead to reverse effects like vomiting, hypertension, nausea, which has significant limited the extensive application of amifostine. Next. So we need a new strategy is required to improve the safety and efficacy of amifostine to improve its therapeutic effect as a cytoprotector for normal tissue. So we use silica holosphere, which is have attention in biomedical imaging and application. In this study, we use encapsulated form of silica holosphere with amifostine to form this kind of new treatment. Next, please. In our research, we designed to prepare oil-water nanoemulsion of hollow silica nanosphere by ultrasonication of oil-water surfactant CTAB and castor oil. Castor oil is chosen for its biocompatible and low cost. Next, please. In this research, we used encapsulated form of silica nanoparticle with amifostine. This will be characterized to determine the hydrodynamic size particles shaped via TEM and DLS technique. This model of nanoparticle has the capability to change unwanted properties of the drugs. Insolubility, biodistribution, renal filtration, immunogenicity, and enzymatic degradation. Next, please. So, by this model, the half-life and the bioavailability of drug could be increased, while drug toxicity could be decreased. This encouraged us to consider whether encapsulated form of amifostine with silicon nanoparticle could be improved drug efficacy and stability. That way, allowing for low injection dose for variable safety profile. The encapsulated form of amifostine using silicon nanoparticle has never been reported in previous literature. Next, please. So prevention or decreasing of side effects of drugs are the major concern in treating patients who force it to take them for long period or permanently. So in this study, we elucidate a new way to get the treatment without falling in new drug toxicity. The aim of this work to investigate the expression of nephrin messenger RNA in target to investigate the protective role of this nanoemulsion encapsulated form of amifostine with silicon nanoparticle as a cytoprotector drug effect against cisplatin-induced nephrotoxicity in male alpine rats. Next, please. In this study, we use 5 groups. No, the previous, no. There is slide, missing slide. Okay, next. Okay, no problem. In this study, we use 5 groups. Cisplatin group, negative control group, carrier. It is nanoemulsion without any drug. Cisplatin group, positive control group, and amifostine encapsulated with silicon nanoparticle without any nephrotoxic model. The last one is encapsulated amifostine with silicon nanoparticle with protection before cisplatin group. In this table, we show the effect of encapsulated amifostine with silicon nanoparticle on serum and urinary parameters. As we see in cisplatin group, renal serum and urinary parameters are affected with this nephrotoxic model, high level in urea, creatinine, total protein, and decreased creatinine clearance, which is come to almost normal level in with our model, and safety of the nanoemulsion model and safety of the drug alone. Next, please. Also here, the stress markers on kidney homogeneity. We see the elevation in the stress markers in cisplatin group through malondialdehyde, nitric oxide, and peroxidase enzyme, which is decreased in cisplatin group, which is returned to almost normal level with our model. Next, please. Here, the expression of nephrin gene in all groups. As we see in cisplatin group, the expression of nephrin gene is decreased, which is normal, come to normal, almost normal level after protection with our model. Next. Histopathology, as we see in figure A, this is show control group, show number figure, P, carrier, normal figure. C is a cisplatin group showing atrophy of glomerulus, congestion of glomerulus, widening of renal space, necrosis for epithelial renal tubules, pyknotic nuclei, interstitial inflammatory cells. And in the D group, it is a model of amifostine encapsulated with silicon nanoparticle shows safety of this model. Next, please. This is the model with protected before cisplatin group injection. Show moderate reduction, cisplatin pathology alternation for focal inflammatory cells, interstitial hemorrhage with thiopigmatic nuclei. Next, please. This is the figure of cow manufacture. Show the podocyte cells lining the glomerular basement membrane. Next, please. Next. This is more close figure for podocyte cells on the GPM. Next, please. Here is an nephrin protein which is guarded for these pores and protect from microalbuminuria. When this nephrin is gene expression is decreased, so nephrin level decrease, so this pore become very wide, lead to microalbuminuria. Next. Thank you.
Thank you for your lecture. Any questions for Dr. Nadia?
I have a question, please. Tell me, how is the lowest concentration cisplatin that you are using in your experiment? Lowest
100
concentration.
150 milligram per kilogram, 3 times a week for 1 month.
It will be in molar, how much?
I couldn't know them now counting them. I don't know, but it is 150 milligram per kilogram every.
Did you study the dose-dependent effect of cisplatin or not?
Again, please.
Did you study
Start.
the dose-dependent effect cisplatin on your
Alone?
experiments? Yes.
Yeah, I studied with these groups.
And its linear dependency, dose dependence effect is linear or non-linear?
No, yeah. It is dose dependent, yeah, because cisplatin, I make many trials for cisplatin alone, and this dose is moderate nephrotoxic model. Some literatures take less, and some literatures take more. So this is moderate nephrotoxicity. That's all.
Very good.
Any more questions for Dr. Nadia?